Sorry, it's just like slides. There's one video. Yeah, this one is working. Okay, and then the next one. Yeah. And this one. Do you hear?
Yes, ma'am. It is audible.
Yeah, so I want the people to hear the audio from the video in this case, and then in this one. Yeah. Okay. Cool. Just go back to talk. At some point, I think I want people to answer things in the chat. So can you enable them to write on the chat?
Yes, ma'am. It is already enabled.
Okay. So should I start then, or?
Leia, we'll wait for other.
Okay.
Maybe some 2 minutes. We'll wait.
Right.
So I think we should start. The participants will completely.
I think we can start then. Okay. A very good evening to one and all. So it's a wonderful time that we all have gathered here, and we welcome Dr. Laia for our talk. Before I could formally introduce the speaker and ask Madhu to do it, just a few minutes I thought I'll share a little bit. So I got in touch with Dr. Laia very recently. So it all happened in the British Biochemical Society meeting. The 60th anniversary meeting was happening last month. It was an exciting time, and we had a wonderful virtual session. A lot of excellent scientists had come over there. So we just started like a kind of a casual interaction. then I came to know the kind of work that Dr. Laia was doing. So I thought it will be really useful for our students to enlighten them with the latest advancements, and moreover, a recent paper in Nature highlights the kind of work. And it's very interesting. The institute where she's coming from is the same institute where penicillin had been purified for the first time by Dr. Florey. So you have also an historic background for this particular institute. So that way, having a representative from that institute, a researcher from that institute, who have carried over the work all through these years, different generations. And Laia is one of the recent generation scientists who had carried over the work. So I think with this little introduction, I'll pass on to Madhumita. So she will formally introduce the speaker, and meanwhile, we'll wait for other people to join. So once the introduction is done, so we have planned the session like this. So the speaker would share research work, followed by we will have a question answer session. And even while as she is presenting, if you have any questions, you can keep posting in the chat, and we will compile the questions, and the question answer session would be moderated by Madhumita and Nasa. Thank you, one and all.
Thank you, Prince, for this nice short introduction.
Hello and good evening. Thank you, sir. Hello and good evening, ladies and gentlemen. I'm glad to welcome you all to today's webinar. Today's topic of discussion is touching the skin of bacteria with a microscope, biomedical application of nanotechnology. We'll also be having a Q&A session at the end of the webinar. So participants, if you have any question, you can post it in the chat section below. It's a great pleasure for me to introduce our guest speaker, Dr.Laia Pasquina Lemonche. She is a research associate in Professor Jamie Hobbs Group at the University of Sheffield. She has done her bachelor's degree in physics and master's degree in nanobiotechnology and nanoscience at the Autonomous University of Barcelona. She then started her PhD in biophysics at the University of Sheffield, focused on deciphering the molecular architecture of gram-positive bacterial cell wall using AFM. After her PhD, she keeps working in Hobbs Group, focusing on understanding how the bacterial cell wall structure changes upon the antibiotic treatment and other environments. Recently, she has produced first high-resolution image of the structure of bacterial cell wall. Ladies and gentlemen, please join me in welcoming Dr. Laia.
Thank you very much for this amazing introduction, and thank you all for attending, and welcome as well on the people that is looking on the live chat in Facebook. I'm gonna now try to share my screen. And please let me know if something doesn't work or if something doesn't look right, and I can stop. And at some point, I want in the talk to be a bit active, and I want everybody to comment on the group chat. And I'm not entirely sure if I can see that at the same time as my talk, but please, if the moderators can translate to me the answers when I post some questions. So the title of my talk, as it has been already said, is Touching the Skin of Bacteria with a Microscope, which is a really fun title, and I think it's really it's very describing of what I'm going to present. And this is a biomedical application of nanotechnology, and as I did a master in nanotechnology, there's many applications to that, but this is more towards the biomedicine and biosciences. So here's the Twitter handle, Laia/Pasquina, and you can contact me there and keep in touch. So to start, I want to just in a few lines what my research is about. So I'm trying to unravel the molecular architecture of bacterial cell wall using atomic force microscopy, and that's, in other words, touching the skin of bacteria. And we just recently posted and published this paper this year, and you can find it in Nature if you look at this title. And then it's very well explained there in detail, but I'm going to explain some of the most important findings that we present in this work. But there is much more other findings there as well. So if you want to look more in detail, you can just look at them. Right, so what's the motivations for the work? So I don't know if everybody knows about this, probably you do. It's the antimicrobial resistance problem. This is a problem where the bacteria are evolving to become resistant to the antibiotics that we used to treat them with very effectively, and the infections among the society are increasing really rapidly. And in specific, the strain that's resistant of Staphylococcus aureus, the MRSA, is estimated to cause 64% mortality than the sensitive strain. So there have been a lot of news and media attention to this, and the worst part is that even if now there is a few deaths per year caused by AMR bacteria, it's estimated there will be 10 million people a year, even more than cancer. So it's our responsibility as a scientist to tackle that. And the way that our lab is trying to help with this problem is trying to focus on the cell wall. So the cell wall of bacteria is very important because it gives the shape of the bacteria, and it's critical for survival. Its interaction, it helps interact the cell with its environment. And basically the main antibiotics group, like the beta-lactams, such as penicillin, target the cell wall. And so the organism that I'm studying is Staphylococcus aureus, or S. aureus. And basically, if we know more about the cell wall, then we can know more about the interaction between these antibiotics and the cell wall. So just a quick reminder why the cell wall is a good target of antibiotics. Because if you look at the difference between bacteria and eukaryotic cells, they, apart from that the bacteria is 100 times smaller, they have a different envelope. So if you look, they have the plasma membrane or cytoplasmic membrane, both of them. But the bacteria have an extra envelope, which is composed by the cell wall, and some bacteria have even a capsule, which even an external layer. I'm going to focus on only the cell wall, which is what Staphylococcus aureus have, because it doesn't have a capsule. And basically, it's a really great chemical target because we can treat the infection in our body with a chemical that targets this structure, because our cells don't have this structure, so they will not be damaged, and it's very specific to the bacterial infection. So how a real bacteria looks like with a transmission electron microscope is this, a TEM image. So you can see the cytoplasm like here, and it's engulfed by the cell wall. And then this cell wall is the chemical model is that it's formed by long glycan chains that I will talk about later. The glycan chains are the main backbone, and then there is small peptides joining the backbone of the glycan chains. But this is just a model based on indirect data and averaging data like HPLC, mass spec, or NMR. So we have no idea how this chemical composition arranges in 3D in the cell wall. And as it's so important, there is a link between the cell wall architecture and the function that at the moment it wasn't known when we started the project, because it's very crucial, but as we don't have a direct way of looking at it, it was an unanswered question. So what do we need to do to answer this question? We need a very powerful microscope to see closely to the cell wall. So this is a 3D storm image. This is a fluorescence microscopy image, and it's the most powerful optical technique at the time, at now. And with this technique, we can see individual molecules, but the issue is that the resolution, it's limited by the diffraction limit of the optical microscope. And I'm going to talk more about that later. So if we want to look at the cell wall, each of these blobs is a cell. So the cell wall will look like this when we zoom in. And you can clearly see that this is not enough detail for us to know what's the structure and how the antibiotic interacts with this. So we need another type of technique. Why is it that optical microscopy doesn't work for this? Why is it that we cannot push more the resolution on the optical microscopy? Well, I'm going to just talk very briefly about the very basic stuff. So what is the principle of vision? How do we see humans, animals, all of us? So there is a light source that has to hit an object, and then the object reflects the light to our eyes that act as a lens and allows us to see it. If the object is microscopic, this is how it works. But if the object is microscopic, so it's very small, we cannot see it with our eyes. So what happens is that the light source will hit, And then we'll have a lens that will amplify this light and condense it and colliding it to the object. Then another lens that will amplify the reflection of the light from the object so that we can see it normally. And this is how an optical microscope works, as everybody has done this already, at least in high school or in university. But this is not useful for every object. So what happens if the objects are even smaller than microscopic? So when the light hits a microscopic object, the light, it's not infinitely small. It has a diameter that's called wavelength. So the wavelength of the light has to be about the size of the object, so that then the objects can basically reflect that light and represent correctly the shape and the characteristics of these objects. However, if we have a nanoscopic sample like DNA or glycan strands or peptide glycan, this is how the light would look like in comparison. So instead of seeing individual molecules, we would see a blur. And this is what happened in the image I showed you before. So if the object is much more smaller than the wavelength, this cannot be used. And this is called the diffraction limit of the optical microscopy. So for smaller objects, there is techniques that people are developing to try to overcome this, this technique, but then there are alternative microscopes. So the microscope we use is called AFM, and this stands for atomic force microscopy. So now I'm going to do a small exercise. It's very, very simple, and I just want everybody's attention for a moment. So imagine that you're in a very light and in a very lit room, and there is an object in the room, and I ask you, what object is it? And everybody will say, well, there's a box. I mean, it's obvious, we can see it. But what happens if now I turn off the lights? And I ask you the same thing, what's in the room? And you have to answer me. You cannot say, oh, I don't know, you have to answer me. So could you please tell me, how would you know what's in the room? Like, I would like if people can try and just answer the chat, just a couple of words. Let's see. And see if everybody touch and feel. Yeah, touching. Great. Thank you very much. So exactly. So it's the first instinctive nature that we have. When we are in a room that's dark, we would immediately start to touch things. People that unfortunately they're blind, they use this technique to read, and they use the Braille technique. So this is what scientists thought. If we cannot see an object because it's too small, we will touch it. And this is the principle of the microscope I'm going to explain. So it doesn't use light at all. It's not a conventional microscope. It's a microscope that uses the sense of touch to feel things. So what do we do when we touch things? How do we know what's in there? We use our fingers to probe around. And the fingers, it's about 1 centimeter big. So the molecules and the materials that we want to look at are atomic or molecular, so they're very, very small. So we need to use a probe that's the same size of the things that we want to touch. So the end of an AFM probe, it's between 10 and 30 nanometers or even less. But roughly, it's the same as our finger, just a million times smaller. And obviously, we have to build a machine to move it. We cannot move it ourselves. So this is how it looks like on the left, a real AFM. And this in the middle is a schematic of an AFM. So we have this little finger at the end that's touching the sample. And this obviously is very small. So how can we know what it's doing? How can we see it? If I've already said that we cannot see these things. So if we put this small finger at the end of a flexible cantilever that moves up and down, and it's fixing one end, and then we can model very perfectly with physics the movement of this cantilever. And we know the movement of the cantilever by shining a laser spot at the back of the cantilever, and the back of the cantilever will be much bigger. So this will be something that can be detected by the laser spot. And then this is shined back to some sort of collector of light. So it's called photodetector, and this will be connected to a computer, and it would tell us the exact movement of this cantilever. So it's very tricky because to make this work, you first have to have a really good cantilever. You have to have the laser spot directly at the right position, and then you have to have all the electronics working. But imagine that everything is perfect. So the way that it would look like is this is a meter long, and this is the machine that controls everything. And here at the very small place, there's a holder where you can put the chip, and the chip, these are normal tweezers, so you can see the chip, you can handle it, you can move it around. And if you can see at the very end of this chip, there is a small hair. So this hair is actually the cantilever, this long part of the cantilever. That's about 100 microns already. So it's around the same size as a human hair. And then at the end of this small hair, there is this spike, and this spike is the Afm probe, and it can have many shapes, but usually it's a pyramid. And at the end of the pyramid, it's about 5 to 10 nanometers on a good day. So with this very precise finger, we can now go and touch the sample. But how do we make it work? How do we build an image? So first of all, we are relying on the physics and the interaction between the sample and the tip. So we monitor this force sensing machine that's called the Afm, and we will see the bending of this cantilever depending on what the sample looks like. And yeah, I just want to point out that this is not. Keep going and see how the Afm moves. It's a visualization on what's an Afm working. So you can see the laser shining on the cantilever. In this case, it's a triangle. And then at the end of the cantilever, there is the pyramid. That's a tip that's touching some sort of coils that are laid down on the surface. And as the cantilever is going up and down, the laser on the photodetector is going up and down as well. So you can map the movement of the cantilever by looking at the laser at position perspective from the non-movement. So then this is basically the principle. This is how it works. Then a bit more detail. What's it doing when you want to do a line? So this is only in one point, but when you do a line, basically the cantilever stays fixed. The end of the cantilever that's fixed, it has a very fixed end. And the rest of the objects on it, it will go up and down and it will bend. So then the vertical position of the tip, it matches with the cantilever deflection, and it matches with the topography of the sample, as you can see. Right, so there are different Afm modes. The one I just presented is called contact mode because you're always in touching with the sample. But for biology, what it was discovered a few years ago is that this mode of contact Afm, it's not good for biology. It's good if you have a metallic surface or a nanometric chip or something like that, that it will not deform. But as you can see, if this is more or less in scale, the tip is massive and it's really big. And if you move something really big on top of a very small molecule or a very small sample that's soft, it will deform or even it will move. So when you use contact mode on a biological sample, it doesn't work. So scientists have to invent another mode a few years back that's called tapping mode or non-contact mode. And basically with this, we're oscillating the cantilever continuously, and then it's much, much more softer because we remove all lateral interaction with the sample. And you can see here the same sample being scanned with the tapping. Is there any questions at this point? No, okay. So keep going on. So then this is what happens when we're scanning in tapping mode. So we start with an oscillation, and we mark this oscillation as our set point. So the set point will be the oscillation that we want the Afm to keep. And then when there is an object, this interaction between the tip and the sample causes the energy to be absorbed by the tip, and then the oscillation gets smaller. And then as it gets smaller, we have to tell the system tries to keep the oscillation as before. So then it has to move the cantilever up to overcome this obstacle and to make the oscillation good as before. And then when there is a fall, it's the other way around. As the tip doesn't have any resistance, the oscillation will increase. And then you have to bring the tip close to the sample again to maintain the same oscillation. So this will look like that. The amplitude of the oscillation changes from low to the same to high to the same. And then this translates to a topography that's this step, that's the same as the sample. So it's a very simple working principle, and it's not hard math at all. It's just oscillation, harmonic oscillations. But we just need to know that it's a very small tip moving up and down on the sample. Good. So this is Video I want you to look at, and it's gonna have some voice, so I'm gonna just shut up for a moment and explain the tapping.
Atomic force microscopy. 1. Finding the surface. The microscope's canopy is activated to vibrate resonance. As it is lowered, attractive forces between the tip and the sample alter the cantilever vibration. The cantilever returns its unperturbed vibration when withdrawn from the sample. The set point for the cantilever vibration determines the distance between the tip and the sample. 2. Scanning principle. If the tip is scanned across the sample, the tip height is adjusted by feedback to give the cantilever vibration set point. The distance between the tip and the sample constant. 3. Scanning the sample. As the tip is raster scanned across the sample, the tip height is adjusted to give the cantilever vibration at the set point, and so the tip follows the topography of the sample. The height of the tip is recorded along with the coordinates of its point in the scan. The resulting map of tip position can then be processed to render an image of the sample topography with nanometer resolution.
Right, so thank you. I hope you had a better idea of how the tip works and how the AFM works, but I'm sure you're all excited to see a real AFM working, and this is the only video available because it had to be videotaped by a SEM. So this is how it looks like. This is the end of the tip. Then there is the surface. When you zoom in, you can see this small end of the tip scanning sort of particle there. And you can see it's really fast. Actually, what happens is that the tip is still, and in this case, the sample is moving underneath. This helps stabilize the laser. So most of the FMs work like this, where the surface is actually the one moving. And it's going line by line to build an image. So when you're actually doing FM, you have to wait for each line to be made. It's not like an optical microscope that you can just take a picture. And I think now it's when they're retracting and they're approaching to the surface again. This is a very complicated process because you can break the tip. And it's actually the real size of the tip at a zoom of 100 microns. And you can see how small the tip is and how close this gets to the surface. And this has to be in an angle so that the tip doesn't touch the surface. So this is the image that they got in this case, in this video, of this particle after the AFM was done scanning it. So it's a 2 micron by 2 micron scan, and the particle is around 200 nanometers. So this would be similar to the images I have. And yeah, we'll see later on. And you can see that by the height, the colors of the AFM images indicate height. So the darker it is, the lower, and the brighter it is, the higher it is. So what kind of things we can do with AFM? We can do lots of things, but here I have just put a few examples of AFM in biology. We can image eukaryotic cells, cancer cells, bacterial cells. We can image lipid bilayers, which are the membranes of the cell. We can image DNA, and we can image single protein. And all of this can be done in liquid, which is great because one of the first things that we wanted to do is to image live cells. And this means that we are not drying them, or we're not treating them with anything. And it can also have another positive, is that it can use the mechanical properties of the material. It can detect them, it can study them, because it's a tip that can be used as a probe and to make forces. So then you have a force scopes, and you can analyze many things like the elasticity and the deformation and the adhesion of the sample. But in my talk, I'm just going to talk about images, but so that you know, it's very versatile in biology. Right, so what are the objectives of our research? As I said, we want to look at a cell wall, and the cell wall have 3 main levels. You can have the shape of the cell wall in terms of cellular level. So there is people studying how much the cell changes. And then there is, does somebody has a question? No, okay. I'll try to answer the question on the chat later. So basically, yeah, we'll have the cellular level, and then we'll have the atomic level, which is studied by chemistry. It is studied by HPLC and NMR and all of this. But the molecular level, it's a mystery. Nobody has studied very in deep because there is no good technique to do it besides AFM. So we're going to focus on molecular level. And then the other objectives as well is to try to study different organisms that are gram-positive bacteria and different mutants to try to understand the differences in cell wall. So here we have bacteria images, purified bacteria of Bacillus Subtilis, Staphylococcus, Aureus, and E. coli. So we focused on these two because they're gram-positive, and as you can see, the Bacillus Subtilis is a rod shape, and then Staphylococcus aureus is a spherical shape. So we wanted to see if there was a difference in structure that relates to shape. And then there's two approaches that we use. We use living cells to study the external surface of the bacteria, and then we use purified peptidoglycan to study the internal structure of the bacteria. So how do we do to image a living cell on the AFM? We first grow the cells in a lab. We put them onto silicon grids. We let them incubate for a while, and then we use the liquid that best helps to get a better image, and we put it on the AFM. And we use the tapping mode, as explained before. And this is the principle. So it starts vibrating, and when it touches the sample, it vibrates less, and then starts the scanning and moving around and altering the vibration, and then that helps the AFM move around and to get the topography of the sample. So we do this on the grid, and we have the AFM tip. We move line by line, and then we build an image like this. These are two bacterial cells, one that was about to divide when we put it there, and the other one that's a single bacterial cell, both of them trapped in individual holes. And we can then even zoom more and look at the structure. So this is already part of the paper. And so when we look at an individual cell, what we see on Staphylococcus aureus is this. So we have two different structures. First, we see concentric rings. They're very ordered, and they represent the most new material that the bacteria has placed. And it's just after the division, this is what it looks like. And then the other part is a more mature cell wall that it's completely broken up, and it's completely random mesh of fibers. So if we zoom in this one, for example, we can see that it's very complex, and it has fibers that are indicated here on white, that they are individual or double glycan strand. As I said at the beginning, the peptidoglycan is made of glycan strands and then peptides. So here, the lines we're seeing are glycan strands. So we're looking at molecules on a living bacteria cell. And then on the rings, we can even see this more clearly. We can see each line very, very distinguishable from the rest, and each line is a glycan strand that has been placed there in this ordered way. And then we can even image along the glycan strands. We can see some periodicity and some structure. This would correspond to the chemical structure of these glycans. So it's called 4 nanometer helical pitch, and it's been studied by the HPLC and NMR community. And here we're just finally seeing it for the first time. So here's a bit more of this. So when we looked at the rings in more detail, we could see this periodicity clearly, and we could even measure the profiles. And then this corresponds to the two hanging out of peptides at the end of each glycan, that they're pointing to the same direction, and then we can see them as bumps. And when we look more at the transition between the rings and the mesh, we could see it was a very sharp transition, because this is when the cell has just divided. And then this is not a progression of time on the same cell. They're in different cells, but you can see that the rings start to break up and start to have holes, and then these start to break up more, and eventually it just looks like a very random mesh. So we also did some studies in terms of orientation, and this is where we found that it starts being very ordered, and then it evolves to be very disordered. And this is important because all the rest of the studies are portraying the bacterial cell wall as a one uniform brick wall, but this is not uniform at all. It has very different areas and it has very different structure. So then we had to do some controls for our experiments. So I have only talked about peptidoglycan, but I have also mentioned cell wall in general. Why do I keep differentiating this? Because the cell wall has two more components. They are called waltecoic acids, that they're attached in the middle of the peptidoglycan layer and they're hanging up from the top. And then there is the lipotecoic acids, which are anchored to the member and they're going inside the peptidoglycan layer. So these other complex polymers, they're there, but they're moving and they're not forming a very tough structure. So what we assume is that we don't see them with the AFM, because when we look at the AFM, we only see the very tough peptidoglycan structure, and these are just like hairs, and they're just like little things that stick out and we don't see them. But we had to do a control, so we used taro, which is a mutant that doesn't have waltecoic acids, and we corroborated that it has the same structures as the wild type. And then we removed the lipotecoic acid using conditional mutant called LTIS with GDPP CAN point mutation, and then it also looks the same. So this was just to verify that wherever we see it's the rings and the mesh, it's definitely peptidoglycan structures and it's not the other components of the cell wall. And all of this is just the external part of the cell wall. But we wanted to also look at what happens on the internal part of the cell wall. So to prepare this, we have to purify the peptidoglycan. We first grow the cells, we then kill them and break them and remove everything of inside. So it's like an empty shell. And then we put this empty bacterial envelope peptidoglycan flat on a surface. And then we can image it with the FM and we can distinguish the external versus the internal structure. And this is what it looks like on the FM. So this is a cell that was broken by this area, and then all the contents were extracted. And this will look like a balloon full of water, and then you break it, and then you just place it on the surface, and it's a broken balloon. And we can clearly see the outside versus the inside structure. So when we do this on liquid, we can also do this in air, but basically we just have to have this saccharic concentration, and then we dilute it and we place it on a surface using some glue, and then we can image it with the AFM, and we can keep drying it and hydrating it, and it will not affect its structure. That's also very interesting. So in this case, we use the same AFM, but use a slightly different technique, and this is called peak force technique, and it's basically using force distance curves. So as I said before, you can have the mechanical properties by approaching the tip, pressing, and then retracting it, and this would be a force curve. But this is also a way to image. If you do this at each point, and then you just move the tip from line to line, you can also get a really good image without any lateral force whatsoever. So we do this, and then we can image, as I said, the inside part of the structure and the external or outside part of the cell wall. And this is what it looks like. So from the external part, it's no surprise that it looks the same as the images that we found on living cells. So we have the ring section, which is the new peptidoglycan, and the mature cell wall, which is the random mesh. We can even have these fragments with the two of them more clearly than in the living bacteria, because the fragments just break randomly. And when we look closely, we just see the same kind of structures. And the important part was when we look on the inside. So on the inside structure, these are all on the same scale. You can see that it's a bit different, and it's a very tight mesh. It's random, but it's like a 2D flat mesh. And the arrows are pointing to individual glycan strands. So you can see them here, you can see them there as well. And each of these lines in this image points to a glycan strand. So this is really good because now we can have high resolution and seeing the glycans in the internal part as well. And then we did a bit more of analysis. So this is something that's really important. We have to do analysis on our images because it's not enough to just say, oh, look how pretty they are. So if we take the image, this is the 100% content and data on the image, and this is the three-dimensional representation of this image. And if you start to slice the images at different percentage, you can actually see different things, and you can look at the pores, for example. So if we look at this slide, and then we take it apart and we like put the pores in black and the rest in white, we can see that it's more clear and it's easier to analyze than having the complete image. So we did this for many images and for more than 100 holes, and we get the distribution of the area of the holes on the external section and internal section. And we did this in a more automated way, in that the program basically slices the images and then analyzes the holes at different slices, and even you can put different slices together with colors to see the depth map of the holes. And you can clearly see that, for example, this hole is deeper than these ones here because it has more layers. And this will be the process from the raw data to actually having the holes and analyzing individual ones. And then the graph that we can build at the end is having the distribution of area of the internal section and external section. And why is it important? Because we can finally have a picture of how a segment on 3D of the bacteria looks like. There is holes on the outside that get smaller as they get through. So the actual limiting step for a particle to enter the bacteria are the small holes on the inside, not the big ones on the outside. And this is really important for antibiotic development, because if they develop an antibiotic that's too big, it will not go in the cell, for example. So this would be how it looks like external to the internal. And we can even look at the pore and have a three-dimensional volume reconstruction of a 13 nanometer diameter pore, and this would be on scale of 10 nanometers. So we're actually looking at a very small and a very detailed way at the cell wall. And so this is also on the paper on figure 2 and extended data 4. And this is basically all the important parts on the Staphylococcus aureus. Then we also were interested as well on the division process. So it's really important to study the division process, because if you can stop the division process, the cell wall will not reproduce and the infection will not advance. So another advantage of using purified bacteria is that you can actually look at these semi-formed septa and having these middle stage basically visualized at different stages. So you have the outer cell wall and you have the semi-formed septa in the middle. And then this is the beginning of the septation process. Then this is a bit more advanced. And as you go in time, you can see the inner cavity closing in. It's not the same cell. There are different cells because they're dead. So they don't progress once you put them on the FM. But the problem with doing this on a live cell is that the FM cannot go and see through. So this is the best we can do, and to have a progression of time and study the different stages, and then even trying to get high resolution on the individual parts. So this is what I did when we looked at the septa in high detail. So we realized the septa is not ordered, and there was many papers in the literature saying that the septation process happens in an ordered way. And it does. It's just that this part of the septa is the one that gets inside the cell. And as I said, the inside the cell is very tight mesh. So it has to be tight mesh here too. But we had to just make sure that we design a program. So I did a MATLAB routine to basically characterize the angles of these glycans to make sure this is a random orientation. You have glycans going in almost every angle. And then when I compare this to the internal cell of the Staphylococcus aureus, it's also covering most range of the angle, and it's also ordered. But when you look at concentric rings, as I showed before, the concentric rings are only going in one direction because they're ordered. So with all of these, we could build this model of how the septa looks like when it's building the cell. So it has 4 different layers, and 2 of them belongs to one daughter cell and the other 2 to the other daughter cell. So they will have rings in the middle, and they will build the cell wall with the rings. But then the other part, the machinery cannot order that material, and it just kind of puts the material however it's laid down. That's why it looks like this disordered mesh on the inside part of the cell. And when the cell splits on the outside, we can only see the rings. So this was a really interesting advancement, that there is these 2 different layers on the septa. So finally, we could build this model of the cell wall of Staphylococcus aureus. So we have at the very new material, we have rings. And this is a model image from this image. So we made this with the computer, but they're from the AFM data. But it's a bit more clear to see them like that. This is a mature peptidoglycan. And then when you look at it, it looks like a mesh. It looks like a sponge. It's much more porous than everybody thinks. And then this is the inside, which looks really, really tough and really dense. And you can see the pores on black here. That the pores are about 6 nanometers. So we have characterized that as well. Then we have the septa. As I said, we have this amazing model of the septa. And then we have also like the section, as I said before, we have the section of what happens when you go from the outside to the inside and the comparison. So we have done some quantification on the images as well. And then the last thing we did was study Bacillus subtilis. So this is a rod-shaped bacteria, and we found it has the same characteristics as staph, except that the internal section, it looks very ordered. So if you can see here, this is as well like the rings. It's a very ordered way because it's ordered on the short axis of the rod, and that allows the bacteria to have this rod shape. The septa otherwise looks very porous and very meshy. And then we also did some measurements on height. So it looks very thin in air, but really high in liquid. So it's a very liquid hydrogel that absorbs water. And then we have as well images on living cells. So this is a living cell of Bacillus, and we can go close and have images of the mesh and the rings. And so finally, we built this model, not just of the Staphylococcus aureus, but the gram-positive bacteria, because they share everything except for the internal cylinder that's a very dense mesh. And I think that's all I wanted to present to you today. So to show how we can use a very new nanotechnology tool to study very in deep a biological species, or in this case, 2 species of bacteria, that they're very important to advance towards the battle against antimicrobial resistance. And this is all. I would like to acknowledge every member in the Hubs group, past and present, and the members of Foster Lab, past and present, and all my the people that is helping us finance this project, which is the Wellcome Trust, the University of Sheffield, and Imagine Life, and the Florey Institute. And now I'm really happy to take any questions. Thank you.
Thank you, Leia. It was a wonderful presentation. Yeah, it's, I mean, a ton of work you have done. So I think people have started posting a lot of questions in the chat. I think like Madhumita, can you moderate? You can read out the questions so we can save some time. So you can start answering the questions directly.
Sure,
sir.
I have already read the first one from Satya. So basically, this is a very interesting question. We don't know what is the molecules that we're seeing with the AFM, because what we only see is the structure, the surface. It's like if you have a mountain map, you don't know what color the mountain is. You just have the map. That's a 3-dimensional map. So there are different techniques we can do. We can use fluorescent probes to kind of correlate that with the AFM. Or we can even use chemical probes, which are basically you put a chemistry coating on the tip that will interact only with a specific molecule, and then you will see that interaction only happening at certain places of the image. But you have to have first an image without the chemical without the chemical coated tip, and then you add the chemical coated tip, and then you can detect some molecules. And recently there is this technique really cool that's called IR AFM, which is infrared combined with AFM, and the infrared can detect the spectra, and it can tell us the chemical composition of the things that we're looking with the AFM. So there's the different things to go around with that. Should I just read the next question, or? Yeah, there is a question by Subisha that says, how will you design a probe to detect bacterial cell or cancer cells or DNA? You can use all of that. You can image all of that with the same probe. So you don't have to design different probes. There is many different probes by different applications, but generally the same probe can be used to to image all of that. You just have to know what sample you put when you're working with it. I think that's all the questions. Right, I have a question from Arvind on what's the difference one can find in the structure of resistant bacteria and the common one of the same species. So this is very interesting. I think this is something that's going to get published soon, but it's something that another colleague of mine is doing in the lab. And we're looking at the MRSA strain, and we're looking at Staphylococcus aureus strain, and then we can compare the differences. For now, the main difference we see is that the cell wall is thicker in the MRSA strain. That's why it's harder for the antibiotics to break it. But we have to look and see if there is other differences that we can find by looking at more strains. But yeah, it takes a long time. So all of these things I presented took like more than 5 years. So it just takes an extremely long time because AFM is a very tricky, very slow technique, but it can be very powerful as well. Right, so
if
any more questions?
If people have questions, you can just post it in the chat.
If somebody wants to talk, I'm also happy for them to talk.
So Leia, you said like you're also working on treating the bacterial cells with antibiotics, and then you would do the AFM work. Yeah, that will be very interesting.
Yeah, it's not finished project yet. That's why I couldn't present it, but it's really interesting, and it's proving
because
that
whole range of cell wall inhibitors work that way. So we would be able to like differentiate how different cell wall inhibitors bind in different sockets of the peptidoglycan layer. Whether you have done like kind of lysozyme treated cells apart from antibiotics, that kind of work have you done?
Yes, we have tried with living cells, adding lysostaffing, things like that, and it just completely destroys it, and you cannot have a proper image because it degrades so fast. So we have to find a way that we can see the process happening because it's a slow technique. So yeah, but it's really interesting.
Yeah. Thank you, Leia. And
sure.
If students have some more questions, you can post in the chat. Maybe we'll wait for 2 more minutes.
And participants, you can also use raise hand option if you want to directly ask question to the speaker.
I will write my email in case anybody wants to write me an email. I'm also happy to answer that.
Sure, ma'am.
Right, so the rendering, somebody asked how the rendering is done. So this is done automatically by the AFM software, and all the commercial AFMs, they have this rendering happening on real time. So when you're imaging, you can actually see the image built up and it's already rendered. So you don't have to do any electronics or any computer things to do that. It actually automatically happens. And then you can alter the image later post-treatment with softwares to make the image look pretty or to have different colors or to have more contrast. But the rendering is already done, and you can visualize it in 2D as most of the images I presented, where the 2D is the X and Y, and then the Z is the color of the image. Or you can just take an option of presenting it in 3D. The problem with the 3D was that you lose the X and Y resolution. That's why we did these models on 3D at the end of our paper, where we used a different approach to have the 3D, but without losing the X and Y resolution. Yeah. So to render, it doesn't take long. As I said, it happens simultaneously. But the image itself, as it's building line by line, you will have to wait approximately 10 minutes for one image. And if you want to do a really high resolution image, you'll have to wait 20 minutes or 30 minutes or something like that. And then it's like you do one image, and then if you want to move something else, you have to wait to do another image and to focus on another place, and it's just a slow process. So a typical AFM can be 8 to 10 hours of scanning to have a few good images that you can use. Yeah.
Thank you, Leia. So,
No worries.
so I think like we can wind up. Madhu, you can just take over.
Yes, sir. Thank you, ma'am, for the wonderful speech. I, on behalf of Anna Study Circle, extend a very hearty vote of thanks to Dr. Leia for gracing your important work and sharing with us your findings and opinions today. I also extend my sincere gratitude to Dr. Prince R. Prabhu for the constant engagement and support. I would like to acknowledge our gratitude to all the participants for being with us evening and making this webinar successful by your active participation. It's been a great pleasure. Thank you very much.
Thank you, Leia.
Thank you. Also read messages.
So you want to say something?
No, I'm just reading the messages and see if there is an answer things. Thank you all for.
Yeah, we have recorded the session and it's available in YouTube, and we also did live Facebook streaming. So people can also check up the link over there. So you can even post comments, and we would be sharing Dr. Leia's mail ID so that if you have any doubts, you can actually pass it on to her directly.
Sure.
Thank you, one and all. So
Thank
bye
you.
for today. Yeah,
Bye. Bye.
Bye.
दिव्यानी है क्या? दिव्यानी इसमें चैट बॉक्स कैसे आता है जूम ऐप पे? डेट हो गया है 26 नहीं तो इंस्टाग्राम यूट्यूब व्हाट्सएप फीडबैक लिंक में दिया है